So it's my pleasure here to introduce the last speakers of this session. The overall theme here is forward-looking ideas in genomics, and we're going to begin with Rahul Satija, who is a core faculty member at the New York Genome Center and has an associate appointment at New York University. And Rahul is probably the youngest speaker. I don't want to say that for sure, but he is a cutting-edge genomicist, interested in both technology and the biology of genomics, thinking into the next generation. So Rahul, thanks for coming.
Hello. Thanks very much, Bob, for the kind introduction and very much for the opportunity to speak here today. It's really an enormous honor to meet so many people here and to get a chance to speak. So I'm going to tell you about a new technology, primarily about a new technology that's really come online over the last couple of years. It's focused on the ability to just sequence one cell at a time with the goal of almost taking the microscopes and the single cell analysis we've used in the past and replacing them with a DNA sequencer. But the truth is, this is really a new technology. It's only been around for the past 2 to 3 years. It's one of the most rapid and fast-growing fields in biology, and so it's been very exciting. But it hasn't yet been used, so much every day so far in a clinical context. And I think that's going to happen going forward. And I'll tell you, the latest and greatest for the technology and how it's going to transform healthcare. I'll do a little bit of that. But what I really want to do is give you a sense of how this field has developed and exploded over the last few years. It's been a lot of fun to watch, and I want to give you a sense of how genomics can rapidly change and transform in such a short period of time, and I think single-cell analysis is a great example of that. Okay, so I'm going to start with a very simple picture. This is a picture of the mouse retina. Okay, so the retina is what we use to process light and transfer that information to our cortex, and you can see right away from this image that the retina is a very complex tissue, right? You can see that just by looking at the different sizes and the different shapes of each of these cells here. And that makes sense because the retina is going to be a very complex tissue. It has to do a lot of things. We have rods and cones in our eyes to process light. They relay information to a huge diversity of neurons, which relates that information to our brains. So it makes sense that we need something, a very high-resolution image, right, like this microscope image, which lets us look at the size and the shape of every cell individually. So in some ways, this is a very high-resolution image, but in other ways, this is a very low-resolution image because we know almost nothing about the genes, right, about the molecular makeup of the cell, the genes that are sort of governing its behavior and its eventual fate. If we want to know something about the genes that are expressed in each of these individual cells here, then we need to do things like RNA sequencing, right, everything you've been hearing about today, like genomics. So this introduces our sort of overall problem here. So let's suppose that our retina, or our complex tissue is like a fruit salad. Okay, and our goal here is to try to understand the recipe. Okay, we want to know what makes up this fruit salad, and we want to do this with genomics. We want to do this with sequencing. But the problem is that whenever we do sequencing, we need to take thousands or millions of cells together to do the experiment. So we're effectively blending all of those distinct signals together until we get basically a large fruit smoothie. And you can immediately see that once we've done all this huge blending operation, it's not going to be possible for us to reconstruct the individual components. So traditional genomics measures a blend of the millions of individual cells that we're processing and can't get to the signal. And what we work on is an alternative idea where instead of blending all of the cells together, we try to sequence or profile each one individually and then computationally say which cells or pieces of fruit, as it were, are like each other. So it's an idea to do things like RNA-seq, but at the single cell level. So the idea is good, right? It makes a lot of sense. It seems like it would be very cool and get us around this sort of blending problem. But the problem is that RNA-seq, as it was traditionally defined about 3 years ago, needed about 100,000 to 1 million cells to work. Okay? And so we wanted to do single-cell RNA-seq, which was 1 million-fold lower than the traditional input requirement. Okay? So we had some ideas on how to do it, and we had some protocols. But one of the, I guess I haven't even told Bob this when I interviewed. When we first started these experiments, I didn't think they were going to work. I thought this was a silly idea. I thought this was so much below the standard for RNA-seq that this was almost a fool's errand. And so I thought, well, if we're going to try this, then what I really want to make sure is that for our first experiment, we pick a system where all the cells are really kind of similar, where they all should all behave the same way, and I want to see that in our data, and then I'll start to believe the results. So actually, our very first experiment, although it didn't turn out this way, our goal was to take a bunch of cells that did the exact same thing as a positive control, as a pilot experiment. Okay? And so we settled on these immune cells that we had studied in the lab, and we thought these immune cells were absolutely identical in their shape and their size in every way. But to even boost the signal, we stimulated the immune cells with a very, very, very strong pathogen. Okay? We hit them with a huge dose of gram-negative bacteria, of LPS. And we know from studying these cells in the lab that when you hit immune cells with pathogens, they evoke these huge, robust transcriptional responses, right? In this case, they simultaneously, or so we thought, would evoke responses to fight both bacterial and antiviral inflammation. So we thought, all right, well, we're going to stimulate our cells, and we're going to sequence 18 of them individually. We'll sequence 18 single cells and see what we get. Now, the reason we sequenced 18 single cells was because it cost us about $200 a cell to do this experiment. This is an expensive experiment at the time, okay? So we were lucky to get 18. We were super excited about it. Now, I'm showing you some of the raw data here, and you don't need to really understand this, but I'm showing you data for a few housekeeping genes. Okay, these are genes like basic things that the cell needs to stay alive, that it needs to do its housekeeping function. And you don't need to understand this plot, but what you can see is that all 18 cells, here in blue, and in the bottom are 3 replicates of 10,000 cells. Everything looks the same, right? So all the cells express high levels of these housekeeping genes, just like you see in the population. Nothing surprising there. But what if we look at the immune response genes, right? The genes that are turned on when we hit the cells with these really strong pathogens. Here we saw something really exciting, because now you can see that the cells look very, very different from each other. And there are some cases where the cells express, some cells express extremely high levels of some genes, and other cells don't even express those genes at all. And if you looked at the populations, if you look in red, the blended average, of course it looks like there's a huge amount of expression because you've blended everything together. But that's masking the fact that some cells were blazingly high and others were off altogether. So this was kind of really shocking and interesting to us. And you might ask, well, why is it important? You know, why do we care? Okay, so identical cells, we think they're identical, and they're actually expressing different levels of genes. But what we actually realized with some pretty interesting computational analyses is that the cells weren't identical at all. We think they probably were identical to begin with, but when we hit them with the pathogen, they responded in very, very different ways. And so some cells expressed very, very high levels of the antiviral response, for example, and other cells had a very, very strong inflammatory response. And all I really wanted you to take away from this very basic, very toy, simple biological experiment with 18 cells is that we could take things that we thought previously were identical, and we could divide them into groups that we hadn't even been aware of based on their gene expression. So let me give you a sense of where we were at the end of 2013. Okay? We could do single-cell RNA-seq on a small number of cells. It was really expensive. We could get a sense that the cells, that even when we think they're the exact same, they can behave very differently. And we can use that information to uncover groups and groupings that we didn't even know existed beforehand. So that was all pretty exciting, but we realized that at 18 single cells and $200 a cell, we weren't going to be able to learn very much. We wanted to do some really cool experiments, not looking at cells in a dish. What about the dendritic cells in the body? Right? We wanted to know, how do the dendritic cells in the human body differ from each other? That might be a really interesting experiment. Dendritic cells, of course, were discovered just a couple of miles from here at Rockefeller University. And since then, you know, about 20 or 30 years ago, we sort of realized there's not just one type of dendritic cell. There's sort of 4 canonical populations of dendritic cells. That's, you know, immunologists have studied these and profiled these for a long time. But of course, they didn't have our single-cell RNA-seq. We thought, well, maybe we'll learn something new if we look using our new technology. But we couldn't pay $200 a cell. So we spent about a year and a half to 2 years, and I won't go into the details here, but building a lot of cool ways to try to reduce the cost of these experiments and to increase our throughput. So version 2 of single-cell RNA-seq is a fully automated and robotic pipeline. First that I built during my postdoc in Boston, and now we have a much more advanced version right here at the New York Genome Center, where now we can sequence 1,000 cells a day, and we're at 2 orders of magnitude lower cost, which is really, really exciting. Right now we can start to think about doing some interesting experiments. So in the context of these dendritic cells, we took 1,000 cells from a human patient, and we did single-cell RNA-seq of 1,000 cells in a human patient. And we expected to find these 4 canonical populations that people have known about for years. And of course, that is exactly what we found. We found the same 4 populations that immunologists have discovered and profiled for many years. But we also found a 5th one, a rarer subpopulation. Each dot in this plot is a cell, by the way, we found a fifth population that nobody had discovered before. And that was really kind of exciting and unanticipated for us. And one of the things that was neat was now what we could do is we could actually say, well, what's different about this fifth group of cells than the other four? What's unique about it, right? And what we actually realized when we put those cells into a dish is that they would divide over and over and over again. In contrast to the other differentiated dendritic cell subtypes, these guys were behaving almost like a dendritic cell stem cell. And we knew that they must exist in the body because we have to replenish our immune cell types every single day. But we never knew what genes they might express. We never knew how to look for them. And so this technology gives us a microscope. It gives us a way to find things we hadn't found before. And of course, not only now do we know that these cells exist, but we know exactly which genes that they uniquely express, what genes give them their stem cell identity, what genes make them precursors. We can use those if we want to develop drugs, if we want to develop targets, if we want to develop therapies. So it's not just discovering the population, it's discovering what at the molecular level makes them unique. Now, this is a very simple example of something where we thought there were 4 types before, and now we think there are 5. Maybe that's exciting, but of course it's not an experiment that we can only do in human blood. We can do this in the brain, we can do this in an animal, we can do this in the mouse, we can do this in almost any biological system where you could apply a microscope. This is a really, as you can imagine, a very basic fundamental assay. And one of the things that's been very fun for my lab over the last year, since we've been in New York, is to profile more than 20,000 single cells. Our robots are running all the time, including right now, sequencing individual cells and trying to find new subpopulations and new signals that we hadn't seen before. So as we closed out the end of 2014 and bleed into 2015, we've moved forward significantly, right? We've had some enormous improvements in cost and throughput. We're down to $2 a cell. We can discover new cell types and states that are present in human beings, which is really exciting, and sort of lends to this vision that maybe one day we'll have an atlas, we'll have a list of all the different cell types and states that are present in a human or a mouse or in anything. That's, I think, really exciting. But one problem is that if you think about, well, if there's only 4 or 5 cell types in something, maybe 1,000 cells is enough. But how many cells do you think we need to sequence to understand the brain? I don't think we could do this 1,000 cells at a time. There are probably hundreds, if not thousands, of distinct neuronal populations that are present in the brain, and no robot is going to give us that answer. No, we needed a sort of a more transformative idea for how we were going to be able to sequence tens of thousands and eventually hundreds of thousands of cells. Another reason why we needed to do that, let's say that we wanted to look at a tumor, and we knew that in a tumor, as you've been hearing about today, maybe only 0.1% or 1 in 1,000 cells is resistant to a therapy or resistant to a drug. How are we going to find those cells? Well, we need to be able to sequence tens of thousands of them. So we've been thinking for a long time about how we're going to dramatically improve our scale. And the answer came earlier this year, and this is a really phenomenally new and fun technology called Drop-seq. It's something that was invented when I was still a postdoc in Boston, and particularly by Evan McCasco and Oni Basu. We were all working in the lab together. And the idea is that when you do single-cell RNA-seq, when we do it in a traditional reaction in the tubes that we use are thousands, if not millions of times bigger than a single cell, right? We're wasting a lot of plastic and we're wasting a lot of reagent. And at the same time, the cost grows linearly. So if I want to profile 1,000 cells, it costs me 10 times as much as it costs to profile 100 cells. And the number of experiments grows linearly with the number of cells. So we had this idea. So the first thing that we're going to do is instead of using traditional tubes, we're going to use these reversed emulsions. These are small droplets of water, okay, that are surrounded by oil and coated with a surfactant. And they're about a nanoliter in size. So they're like a millionth the size of a standard molecular biology tube. But we can make tons of these things. We can stack them on top of each other. We can make millions of them. They're stable. You can put them in the fridge for months. They're basically like little reaction tubes, but they're just very, very small, okay, about a nanoliter in size. It turns out it's actually very easy for us to put cells in these guys. Not to get too detailed, but we use a trick called Poisson loading. So not every droplet, as you can see, has a cell, but it's very unlikely that any droplet is going to have more than one cell. So we effectively have isolated the cells inside of individual droplets. But now, how are we going to know which RNA or which genes came from which cell? We need to put some sort of barcode on the cell, okay? So I'm not going to go into the details of how this barcode works. Essentially, it's a small bead. But basically, it's a barcode. It's like a social security number or a UPC code on a grocery item so that you can mix everything together. But in the end, as long as you have those barcodes, you can tell which RNA came from which cell. So as long as we can get one cell and one barcode together in a drop, we're good to go, okay? So I'm going to show you how we do that. And then after we do that, by the way, we can mix everything together and just make do one experiment and then use the barcodes to tell the cells apart. So this is a movie from my lab. This is Drop-seq the movie. All of the action is going to happen in this little junction right here, okay? So what you're going to see is you're going to see these barcoded beads, these beads shooting in from this channel over here, all right? And you're going to see cells shooting in from this channel over here. They're going to meet in the middle, and with some probability, you're going to have one cell and one bead together, and they're going to go off and form a droplet. Okay, so can we start the movie? Oops, go back. And you need to click, yeah, there you go. Okay, so we're running along here, okay? This is in real time. You can see the barcoded beads shooting through this channel. If we scroll over, we can see these beads coming in and shooting through. If we scroll over just a little bit more, now you can see the individual cells. These guys are actually Drosophila neurons, although it doesn't matter. You can barely see them. These little balls that are coming through and then shooting through the device. They meet back up, okay, here with the beads, and then they go off to form these little droplets. And if we scroll over to the outlet chamber, you can actually see the droplets being made in real time, and then we put them into a tube. And we're making, basically millions of these droplets per hour. Not every single one of them has a cell in it, so our actual throughput is about 10,000 cells per hour, which is really quite exciting. Okay? So now, what's our, oh, sorry, I should just mention that, of course, the real beauty of this experiment is now that we've barcoded all the cells, we can pool everything together and do one experiment so it doesn't scale, right? If I want to profile 100 cells or I want to profile 1,000 cells, it's still just one reaction. And so now our cost has dropped from where it used to be 2.5 years ago at $200 a cell when we were doing 18 cells to now we can do tens of thousands of cells in a day at a cost of less than 5 cents per cell. This is really an enormously fun and dramatic improvement. So how do we demonstrate this technology? Well, I'll show you our published work. We went back to the retina. Okay, this is the same tissue that I showed you before, and what I showed you was that the retina consists of a huge diversity of different cell types and states. But traditionally, we estimate that there are more than 100 different cell types that are present in the retina, although the only way that we can even estimate that is based on their cell morphologies and based on their shapes and sizes. We don't know very much at all about the molecular profiles, about the genes which mark each of these cell types. Until we run Drop-seq, we ran 50,000 cells across 6 biological replicates. And if we look at the results, this is in the same format as I showed you our dendritic cells from a while ago. Now we can see a huge number of distinct clusters, each of which represents a distinct cell type that's present in the retina. So even if we're conservative, we can say that we can call about 37 distinct clusters. As our computational methods get better and better, we realize that we might be even underestimating this by a factor of 2. Okay? But of course, what's exciting is not that we just discover a cluster. Again, we can discover for each of the clusters we have RNA-seq data, we can say what are the genes that are uniquely defined in that cell type. And we can make these, I call these checkerboard plots, because you can see that every cell type we discover has tens or hundreds of genes that uniquely define it. So this is really our fruit salad, right? These are the individual components to our original bowl, and that's very exciting. Except, of course, they aren't pieces of fruit, right? They're cell types. They are exactly what Cajal looked at under the microscope, quite a long time ago, and deemed to be individual cell types. Of course, he did this just by looking at their morphologies and by looking at their stainings under the microscope. And what we're hoping is that as this technology matures, the Illumina sequencer is going to become a very complementary tool for single-cell analysis, essentially a molecular microscope. Okay? So this has been a really, it's been an amazing few years to watch this technology grow from something where we could just profile a few cells in a dish and nobody was that interested by it, to something that I think is genuinely going to have an important impact on human health. I want to point out that this is not unique to single-cell genomics. There are many, many technologies, including genome editing, that have taken off over the last few years, and there are experiments that we can do now that we didn't even dream were possible 2 or 3 years ago. And there's probably a whole bunch of experiments that we're going to be able to do in 2 or 3 years that we couldn't dream were possible now. But in the Genomics Frontier session, I hope you can get a good sense of that. I think one of the nice things about finally now being able to do these huge numbers of cells is we can start to ask interesting questions about human beings. We can ask interesting questions about what goes wrong in disease. And so I'll give you 2 brief examples of things that we're interested in my lab, although, as I said, I think this is a very general technology. The first is you've probably heard quite a bit today about tumor heterogeneity. Tumors are not just one cell type repeated over and over and over again. There's a huge amount of diversity in a tumor. And this matters quite a bit for treatment because, for example, if we attack a tumor with a therapy or a drug, sometimes it works extremely well, but there's a small percentage of cells that remain afterwards that are resistant. And maybe single-cell RNA-seq could be a technology to identify those cells and help us target a secondary therapy that would attack them directly. And so the ability to find these resistant or stem-like cells is a problem that many people are excited about for single-cell analysis. I'll give you one more example that I think is really cool and something we're working on quite actively, and it has to do in the context of autoimmune disease. So when you have a flare in a disease like rheumatoid arthritis or lupus and you sense inflammation in your body, that's a very, very complex signal that represents the blended signals of many, many different cell types, right? It's not just one type of cell that's causing the flare. Each of the different cell types in our body is contributing uniquely to that. And because we're blending that all together, we can't design effective therapies or effective targets to try to get around that pathogenicity. So these are the types of problems that I think single-cell RNA-seq will be able to address in the future. But for now, thanks very much for listening, and I'm happy to take any questions.
Time for questions, and you saw it here first, Drop-seq, the movie. So questions? I have one, I'll start. How do you deal with the issue of going back and forth between the mouse and the human?
Yeah, so one of the things that I think is really amazing about single-cell methods is the ability to do more and more experiments on human beings, not on human beings, but to use humans, to use human tissues. So, for example, many of the experiments that are done in mouse are because you couldn't do things like single-cell analysis in humans. We have to use mouse models because there's no other way to profile human tissues at this kind of resolution. And I think that these technologies can be helpful in trying to get us to do more and more experiments in the clinic rather than having to resort to mouse models. I do hope it is a general phenomenon that the results that we found in the mouse are applicable to the human because that enables us to test drugs and to test therapies. But I think one of the things that's exciting about these technologies is you don't need a lot of cells to begin with, and of course it's single cell, and maybe that makes it more amenable to human analysis. Yeah, so that's a really fantastic question because essentially what we're measuring here is gene expression. And so how do we correlate that with either the anatomy of the cell or, more importantly, the function of the cell? So correlating it with the anatomy of the cell may be an easier question because there are ways, not particularly using Drop-seq, but with other experiments, where we can take a picture of a cell and then sequence it. And that would enable us to look at how its gene expression actually affects its size or its shape. Trying to understand the function of the cell based on this gene expression remains a challenging problem and maybe the source of a longer discussion.
When using Drop-seq, you are taking a snapshot of the cells, the gene expression of cells. Is it possible to combine with some other barcoding system to track the cells over time?
Yeah, that's a really good question. So with all these technologies, we're taking a snap. We're not able to watch a cell over time, right? We're sort of looking at what the cell was doing at that exact moment. The problem is, whether it's Drop-seq or any barcoding strategy, is that when we try to sequence the RNA from the cell, it is a pretty destructive assay. So essentially, we're killing the cell. However, you can imagine doing some very interesting things computationally. So let's suppose that we looked at a retina, for example. This is something we're doing. And instead of just sequencing a snapshot of one time point, we sequenced a series of time points. Then it becomes a very interesting computational question to say, if I look at all the clusters that I find on day 5 of the embryonic time course, can I connect them to the clusters that I see on day 6? So I'm not really sequencing the same cell and watching it over time, but I'm sequencing populations and trying to connect them together. And that's something that my lab is very interested in.
So with this Drop-seq, how many cells you can put together? 10,000 or 9,000 something?
So the limitation now is how much can you afford to sequence? Because as you can see, it doesn't scale with the the number of cells that you put into the experiment, right? But of course, as you add more cells, you have to sequence them more deeply. And right now, it costs us about, let's say, 25 to 50 cents to sequence a cell. So it just depends on how many cells you need and how much money you have for sequencing.
I have a question, also methodological. How do you know, to go back to your analogy with the fruit, a bowl of fruit, how do you know it's actually fruit and not a fruit fly or a piece of hair they got in? In other words, with such small numbers and sort of n of 1 with each cell, I mean, obviously if there's a whole separate category of cell, I can imagine, but as you get in at the margin, you're trying to look at function, especially where RNA is so evanescent, there's so many things that can influence RNA. Could it be the position, inside your device? Could it be the duration between the time it got it, you took it out and put it in the device and so on? So how do you validate the finding?
Yeah, so there's two really good questions there. The first is, how do we make sure that we're profiling a real physiological signal? So with all the experiments that we showed here for drops, we tried to make sure there was a time lag of about 15 to 30 minutes between when the cells were taken out and when they were put into the device. And so we hope that that minimizes any changes in gene expression. But the second question is, how do you deal with these sort of N of 1 problems, right? Like there's some cell looks a little bit weird. And I think when sometimes you only have 1 or 2 cells that look a little bit weird, it's hard to know for sure that you're onto something. And the only way to get around that is power in numbers. So if you need to find a cell population that's present at 0.1%, 1 in 1,000, you can't just sequence 1,000 cells and hope to find 1. You need to sequence 50,000 cells and hope to find 50. And that has profound implications on how how many cells you need to sequence in a tumor, and these types of very interesting questions.
Were you able to identify one or more stem cell-like populations for retinal cells development, similar to the other Pearson correlation chart?
Yeah, that's a good question. So, I mean, in the blood, we know that there should be stem cells present in the blood, and in the marrow, of course, because we're regenerating our blood cells every day. The retina, I don't know if we're regenerating our retinas every day. I kind of hope not. So I don't know if we'd expect to see stem cell populations that are present in the retina. I don't think we found one, or we have found one so far. But I think if we were to look earlier in development, we were looking at relatively differentiated tissues. If we were to look at earlier time points, we might be able to see this.